Dear audience, please welcome the popularizer of science and moderator who will guide us through today's event. He currently works at the Institute of Neuroimmunology at the Slovak Academy of Science, but has received various degrees in neuroscience and physics from prestigious foreign universities. Above all, he values curiosity, which continues to drive him to approach the question of the origin of life in a joint discussion. Welcome, Tomáš Hromádka.
Good morning. Ladies and gentlemen, let me welcome you here at the Pavel Orszag Hviezdoslav Theatre in Bratislava, Slovakia. Let me also welcome everyone who might be watching us live online on the live stream. And good morning, good afternoon, or good evening to anyone who might have stumbled upon us at some point in the future. We are here today as a part of the Asset Science Awards program, the prizes that recognize outstanding Slovak scientists working in Slovakia. And these prizes, these awards are sponsored by Asset and the Asset Foundation. And every year the head or the chair of the selection committee is a recent Nobel Prize laureate. And this year we are honored to welcome among us Professor Ada Yonath, who received the Nobel Prize in Chemistry in 2009. Today, here, during our gathering, we will mostly talk about ribosomes, these magnificent molecular machines that make proteins. And by making proteins, they create everything, essentially everything that's alive around us, and including us. We will also touch upon a related topic of how ribosomes can actually help us or bring us toward understanding of the origin of life. And on the more practical side, once we move from 4 billion years ago to present, we can actually touch upon the subject of how understanding of ribosomes can influence our thinking and design of antibiotics, and how these antibiotics can actually help us fight infectious diseases. Our main guest, our main star of today, is an exceptional scientist and a scholar. She comes from the Weizmann Institute of Science, where she heads where she's been directing, I think, the Kimmelman Center for Structural Biology and Assembly, or Biological Structure and Assembly, and for decades now. She's a legend in the fields of structural biology, biology, and chemistry. Ladies and gentlemen, please give a warm welcome to Ada Yonath.
Ada Yonath is a world-renowned biochemist and 2009 Nobel Prize laureate in chemistry. She is known for her research into the structure and function of ribosomes, which has led to informing our understanding of antibiotic resistance and the development of new antibiotics. Professor Ada Yonath works at one of the most prestigious scientific institutions in the world, the Weizmann Institute of Science in Israel.
So, Professor Yonath, Ada, if I may call you that, welcome to Bratislava.
Thank you.
I hope you had a safe trip, a nice trip, an enjoyable time.
Of course.
Of course. So you attended the gala. I can see so I read about you a lot, right? And one of the interesting headlines that I read was the one that stuck with me, and I will remember that also almost forever. It said, Ada Yonath, the cheerful chemist. And I found I just found it, and I thought it was a very, very nice headline.
Thank you.
So welcome. Thank you for being here in Bratislava. Is this your first time in Bratislava?
No.
No, you've been here before.
Yes,
Excellent.
Many years ago.
Excellent. And I do hope you will come back at some point, because we will be glad to welcome you back at any time.
Thank you.
So we will talk today about something you probably have heard about. So I will start with a question that you probably haven't heard at all during your life. What is a ribosome? Can you tell us? I mean, is this
I heard
thing?
Laughing in the audience.
Yeah, no, there's no audience.
Do you try to make jokes of me?
No, of course, I would never dare.
The ribosome, or ribosomes in general, are intracell chemicals, let's call it, bodies that know to read the genetic code and accordingly to create what the cell needs for functioning, for growing the proteins. So it's a molecular machine that knows to read, to understand if she wants it, and to make proteins accordingly, not too many, hardly any mistake, and in a fantastic speed, unbelievable speed. I can give you an example. I was a very good student as a student in the university, and I was asked to make one bond between two components of all proteins are called amino acids. I was the first in class in terms of time. It took me 6 hours to make one bond. And luckily, I didn't make a mistake. But in the book, it said the mistakes are allowed. The ribosome makes thousands of them in a second and hardly any mistake. So I needed 6 hours for one bond. Ribosomes can make thousands of bonds in seconds. Not in hours, not in days, not in even minutes, in seconds. Now you know what is the ribosome?
Yes.
Almost.
Thank you, almost. So it's a very big, huge, monstrous macromolecular complex in a cell. But if I say monstrous, like how big is it? What can we imagine? Yeah, I know this is probably something like we can see the artistic rendering of a ribosome behind us, which does it resemble a ribosome, by the way?
It resembles parts of the ribosome.
Ribosomes, thank you.
And the connections are not very strong here, but nature takes care of itself. It doesn't need. We enjoy art very much. Don't think that I'm against art or against this. But nature does it a little bit more compact and much more efficient.
So I think the ribosome, because it makes proteins, is essentially one of the most fundamental organelles in a cell, right? Because we can say that essentially every molecule that's in a cell was either made by the ribosome or made by enzymes that were themselves made by the ribosome. So I think this is what makes them the really fundamental parts of everything that's alive.
That's correct.
Right? Yes. Thank you. And I was not trying to make any fun.
That's correct. It's a fundamental cellular functional machinery.
Yes. And it's in fact a machine, right? Because it needs energy and it moves things and creates stuff, I have to say. It actually turns the code, the genetic code, into biological reality.
Correct.
Right? So how does it do that?
Oh, you want to know it all now?
Yes, I mean, no, they're not here. That's just.
You want to know it.
I want to know, yes, exactly. I'm ignorant.
Well, as it is functioning in cells the way we understand, is that they can associate the genetic code to them, the genetic code that brings the information about the sequence of components of proteins. Proteins are needed for almost every function of cells. They can produce the function. There are proteins that are functioning in human cells, in flower cells, in cockroaches, in fish, whatever the organic molecule needs, it makes. It can get the information from the genetic code. The genetic code tells which is the sequence of components that make the proteins. They are called amino acids. This gives the proteins that come out the specific structure that allows for the function. So they actually read the sequence information makes the bonds between the components according to the sequence information. This way they create the proteins and they let them come out at the situation that they already can protect themselves from falling apart or disintegration. So they are doing all of this in seconds. And correctly, and when it's needed.
And how many of them do we actually have in every single cell?
And it depends on the cell and depends on the level of evolution, of the creation of the
complexity of the cells.
Yeah, but also the creation of the organic material that needs them, which animal, which flower. There are cells that have millions of ribosomes. In the liver, 6 million in every cell. 6 million ribosomes are making proteins. Well, not exactly 6 million, but between 4 to 8 million.
Let's settle on 6.
This is more or less the average. But even in cells that hardly grows, there are hundreds, actually thousands of ribosomes. They are not functioning all the time. They are functioning where they are needed. They are made of 2 subunits, 2 parts, what we call the large part and the small one. Both of them, in molecular terms, are very large. And each of them, each alone, the large or the small, cannot do anything. They worth nothing. But when they come together correctly, they can produce the proteins. And they come together when it's needed. So they also know to read or to accept signs. Now come together and make a protein.
So essentially, these are the very dynamic, let's call them 3D printers, nanomolecular scale 3D printers in a cell just riding around.
Yeah. But I think they were built before printer was built.
Yes, of course. They were made, we will get to that. They were very, very long time ago, yes. And they're actually not 3D printers, they're essentially 1D printers that create a string of amino acids put together by peptide bonds, and then those will fold together.
Yeah.
So, not much was known about the ribosome, 50 years ago, right? These were usually the blobs that the molecular cell biologists describe on electron microscopes, right? The Pallade granules, right? But then you came along, right? And you decided to study the ribosome.
I wasn't the first that decided to study the ribosome. I wasn't the first. There were several. I'm not sure you want to hear all the names now, but I'll give you an example. There was a whole Max Planck Institute in Berlin that was devoted to study ribosomes. Max Planck for molecular genetics. They were also in England, in Britain, in Oxford, and mainly in Cambridge, and in the United States. I won't talk about all of them. I'll just tell you, they all wanted to understand how life starts, or actually how ribosomes produce proteins. So proteins have a sequence, the sequence of their components, amino acids. This sequence is the gene that codes for each protein. All these genes together, genes are stored on DNA. All this DNA together sits in the, let's say, the center of the cell or the nucleus, or in other components. And when a gene is needed, this piece of the DNA is being copied to a messenger that knows only this copy. And this messenger, which is made of RNA, is coming to the ribosome, or the ribosome comes to it. A ribosome is being built along it. It means the small subunit and the large subunit come together, and then proteins can be made. Now, messenger RNA was hardly talked about. People in the streets, not scientists, not cellular scientists or biologists, hardly knew about it. They knew genes, they knew proteins. When did messenger become popular? With the pandemic. with COVID. This is the piece of cell of the virus that is being used for how do you say it?
For creating a vaccine that actually
for the vaccine.
Template for the
Thank
antigen.
you. So you know what happened to me about this? When I was talking about messenger RNA, mRNA, which is called in short, people were looking at me, oh, this crazy woman. But now I'm popular. I'm part of COVID.
Because of the messenger RNA.
Yeah, because of messenger RNA, mRNA. So messenger is on the way between the gene and the protein. It comes out of the DNA. The DNA must be double helical. It means they are coming one interwoven into the other. Messenger RNA can be isolated, just one chain. And ribosome knows to read, to get the information from single chains. Therefore, messenger is important. Actually, it's produced all the time in all the cells in order to make proteins.
Yes, it's essentially the language.
Not only vaccines.
Yes, exactly. No, everything. Everything that actually makes us alive.
It goes through the messenger.
Yeah,
now messenger is much more, as I said earlier, much more popular. But in life, it is needed all the time.
Yes, and it's the string. It's essentially the string of nucleotides that actually keeps the genetic information that is then translated by the ribosome into the language of proteins.
So in this model, you see a line going from one side to another, just one line. This is supposed to imitate the messenger, much too long for ribosome needs only a little part of it, but it doesn't matter. This is more or less the way that artists see the translation of the genetic code.
Yes. And there'll also be another tunnel when the new protein is actually sticking out.
Yeah, but there is hardly any place for new proteins.
But that's fine. It will be fine as a model.
Yeah, that's okay.
Artistic rendering, yes.
Artistically, it's very nice. And thank you for making it.
But then let's imagine this is about 200 nanometers big, right? Which is very super tiny, teeny tiny thing for us, right? It's huge for a cell. Now, your task was, let's see how that thing looks like, right?
Yeah, this is
how would you go
a very
about
small
that?
part.
Yes, it's a very small part, but even the small part was actually big compared to other proteins or peptides we knew the structures of.
That's correct. When I was a student, the first structures came out. I'm sure you all know about DNA, the double helix, but this is not a protein. These are nucleic acids.
Yes.
The first protein, I think, was hemoglobin.
I think so.
In Cambridge, oh God, I will remember in a minute. And another protein that was worked out at the same time was lysozyme in Oxford by Phillips, who came half a year too late for getting the Nobel Prize. The Nobel Prize was given to sort of earlier. But anyway, this is what was known. I think there were about 5 or 6 more proteins in the works when I was a student, but the structures a very small one from Dorothy Hodgkin. I'm very bad in names, so I'll remember in a minute, but it doesn't matter. This is what was known. That's all, 5 or 6 proteins. And there are millions of them. Not only millions between different types, but even within cells.
So why do you think we need to know the structures of proteins?
Well, first of all, it's very beautiful.
Yeah, thank you. That's always a good reason, yes. But
That's very important, I think. But mainly in order to understand their function. So each protein does something. One transfers oxygen from the lung to wherever it's needed, this hemoglobin. One is cutting dead proteins, so they will not be contaminating. One is making proteins in order to read or to produce any type of reaction. So they all can do their jobs only when they have the right structure. Even we need structure. We can do it with our hands more than other things that our ears are doing. So we need structure. But this is structure in a global way. Each of our cells have to produce many structures, and they are and each of them is responsible to a specific function, and this is determined by the structure, by the chemical structure.
So it's essentially essential for our understanding of what's going on.
Yeah, if you want to understand, there are people that are happy without this, it's fine.
But I think it's an important point because, at least from my experience, we talked about it briefly, most of biology is based on describing things. Very detailed descriptions, which not necessarily mean understanding.
That's correct.
Right.
And there is very good biology.
Yes, I'm not saying it's a bad biology, but it's incomplete.
Only on description, mainly diseases and things like this. And not everything has to be understood to the very defined point. But if one wants to understand or to develop, like let's say in medicine, to develop therapeutics against specific protein or specific disease, then one has to know the structure. But mainly to understand how this particular protein works, how it is part of a chain of reactions, and how the cell can live or cannot live without it.
So, but then the question remains, right? You have this protein. You cannot just take a picture of it, right, to understand it and to determine its function, right, and how it does things. So
well,
how do you do that?
Today's picture of something is something very, very broad. It can be at many levels. But actually, we want to know where is the position of each atom that makes this protein, and this means to determine the structure. And determining the structure of proteins, of materials, all materials, even salt, everyone, is possible by crystallography. Today there are more advanced procedures, but the idea is using the possibility to make crystals and diffract X-rays through them. Now, diffraction is physics, and many biologists and chemists don't understand physics. But they know that if a beam passes through something ordered, very well ordered, it will be diffracted according to mathematical laws, which are called Fourier synthesis, Fourier laws. Fourier was a French scientist, mathematician. That found the Fourier series of diffraction. If we can measure the diffraction, its position, its strength, and its relation to the other pieces of the diffraction, we in principle can combine them all and get the picture of the material that produced the diffraction. For this we need, I'm not going to go into Fourier mathematics, but we need lots and lots of repetitions. This is why we need crystals. It's not enough to look at one molecule, but they have to be ordered in 3 dimensions, and we have to collect the 3 dimensions Fourier diffractions. And then you have to know mathematics or to rely on somebody else that knows mathematics and combine them together. If we didn't make a mistake and the crystal the periodicity was good, we're supposed to get the structure. And that's not always happening. Mistakes can be made. The order can be less optimal, but most of the times. So when I started, there were 3 or 4 like them. Depends how you call the 4. Now there are hundreds of thousands of structures.
Yes, but then when you say it, right? We just fire an X-ray beam at the crystal, right? And then we take a picture of the diffraction, and we reconstruct the structure from that picture according to mathematical laws. So many pictures according to mathematical laws.
In all directions.
Yes, but then how do you get those crystals? Because I think that's, for me at least, when I was learning about structural biology, that was the magic, right? People spent a lot of time.
This is the magic part.
Yeah.
So science is not only straightforward diffractions or relations or formulas. Science is also some magic, and the magic is to make crystals. Now there are ways that crystals are made normally by nature, like sugar. When you buy sugar, there's little shiny things, they are crystals, or salt. Salt crystallizes on its own just by drying. But proteins, if you dry them, they are gone. They have to be in usually water, but some type of fluid solution. And these have to be arranged in 3 dimensions repeatedly. That's not so easy and hardly happens. Naturally, I think maybe hemoglobin, if you let it stand. I think, as far as I remember. But otherwise, no. If you let the protein stand, you get a hard-boiled egg or cold-boiled egg.
So, but then, when you started thinking about deciphering the structure of ribosome, what did people tell you?
Yeah.
I mean, because
first of all, I didn't start with ribosomes. I was much more careful and humble. I wanted to understand each protein that we know what it makes. I wanted to understand its structure. Yeah. And there were some techniques like making proteins crystallized by adding the materials that are connected to them in a way that is partially periodic. So I wanted to understand protein biosynthesis. It means the translation of the genetic code.
I'm sorry, that doesn't sound like a very humble and safe project itself.
No, safe it was not.
Exactly.
It's still not.
Still isn't.
I knew that there were very strong groups in the world, at least 5 or 6 of them, that tried to understand ribosomes. It means to determine the structure, and failed. So I didn't think that I am good enough even to fail. But I started with trying to understand how to put the spark in ribosome. It's called initiate it. So there are initiation factors that initiate the production of ribosomes and the function of ribosomes. They're called, as I said, initiation factors. In bacteria, there are three of them. At the Weizmann Institute, there was a professor, there still is, Michel Ravel, who worked on this understanding the biology of these factors. So we made a collaboration. He will try to understand, and I will try to crystallize. Ladies and gentlemen, we failed. Until now, I think now there are beginning of understanding the structures of the initiation factors in bacteria, but it's much more interesting how these initiation factors tell the ribosome, start working or start producing yourself. It means large and small subunit. This we wanted to understand, and I failed. I failed, and you know, my fellow scientists, they were laughing at me. They said I will never be able to do it. Other people, more experienced, I was just beginner, maybe 2 years in science. One of them was really, really ugly person. I won't say the name. He took crystal. I was crystallizing in the cold room. He took crystals of sugar, put in water in the bottle that wrote my name on it, and he said, "Here, you have crystals." So this was more or less the feeling at that time that even crystals cannot be made. And as I told you, I couldn't make them. I was fired because I couldn't produce results, or I did produce results that were much simpler and another protein and much slower. So slow and very little information. I was fired.
Yeah. But I think that was, obviously we now know that was a very short-sighted firing, right? But then I think that for me, the important thing is that from the very beginning, you were able to identify the right burning questions that are worth answering.
The question was very good. The person that wanted to answer the question, means myself, was considered maybe very good, but not successful.
Yeah, but the question, the ability to identify the right question is what separates the exceptional scientists from the run-of-the-mill scientists who usually complain that they don't have enough equipment, right? Because usually the way it goes, right, we have this equipment, what can we do with this, right? When you have a hammer, everything looks like a nail, right? But it should be done the other way. Like, this is an interesting question. How do
I make a hammer that fits it?
Exactly.
So I will just answer your previous question.
Thank you.
So, actually, I was fired but rehired under conditions that were not very popular, and not actually I was the only one to have them. I can work, I can even ask for grants, but I cannot be promoted. It means that's it. I shouldn't have any dreams about promotion. I could work. Ladies and gentlemen, this was the best, most fruitful time in my life. I didn't think about any promotion, just about science.
Exactly. There's no pressure.
No pressure.
Just keep going.
And I went for sabbatical, and during the sabbatical I went to a meeting, and the meeting was about protein biosynthesis, still hardly anything known. And it was in February. Most of the people wanted to ski, so they came with ski clothes already to the lectures. They started at 7 in the morning until 11, until the ski time. Only one participant came with a tie and a suit and gave a very, very detailed lecture about how ribosomes are built. And his name was Wittmann, Professor Günther Wittmann from Max Planck in Berlin. And in his last sentence, he said, "My wife," it means his wife, "has sequenced initiation factor 3. So I thought I have to talk to him because maybe the sequence will tell me how to crystallize. But he was too elegant and too European, let's call it. And
Dare we say too German? No, probably no.
I didn't say German.
No, you didn't. Nobody's watching.
But in the last day, I got my guts and I asked him, and he was very, very, very happy to find out a person that wants to understand ribosomes, which from the structure, which was not very popular because it was already known that crystals cannot be obtained from ribosomes. And he invited me to talk to the initiation factor group in his institute, and I came there. I don't want to go into all details, but we decided to start collaboration on initiation factors sometime in August, I think, or so. Mm-hmm. I had a bicycle accident at the end of this winter, and a brain concussion. And it was very, very, very severe brain concussion. This brought me to where I am now. while I was
Please
recovering...
do not take that as a guideline for your future career.
No, you
Might
don't
not
have
work.
to repeat every detail, but only the important ones. While recovering, I couldn't go to Berlin to work on the initiations. So I came late. It was almost Christmas. And they went for their Christmas vacation. I was there for a week or two, and I was very weak. So I sat in the library and read. They had hardly anything in English, mainly brochures of travel companies. And in one of them I read about the bears of the North Pole. You all know that in the North Pole, the bears are sleeping in the winter, hibernating. You sure know, and if not, you know it now.
Those are the ones that eat honey. They don't eat the other ones. Yes, the white ones. Okay.
The white ones.
Yes.
I'm not sure I remember the color, but I remember the hibernation. And
The brown ones.
a group sent by this, or interacting with this travel company, said that they looked into the cells of the hibernating bears, and they found that their ribosomes are organizing in the inner side of their membranes of their cells, almost as crystals, repeatedly. Only monolayer, not three-dimensional crystals, not perfect, but they are packed very well, repeatedly. So I said, here, this is the clue to get ribosome crystals, these bears. But first, I have to think about how to make it 3 dimensions. And second, why the hell it happens? Bear goes to sleep in the winter, and his ribosomes become semi-crystalline. So I thought about it, and I thought that this is the way that the bears are preserving the activity of their ribosomes. So when they get up at the end of the winter, they have to function. They need ribosomes. If they don't preserve the activity, the ribosomes will fall apart. This is what I knew about ribosomes. So under pressure, ribosomes tend to make packed
tight, well-organized
organization. So do you think that I wanted now to go to the bears, to kill bears in the North Pole and start to get the ribosomes?
No, please say no. You did.
This was the answer. I said no, by no means to go and kill bears, even for ribosomes, no. But looking at other system under pressure, this I could.
Yeah.
And I also found a system, a bacterial system under pressure, which seemed to be first easier than a animal system, and second, just bacteria. And this is in the Dead Sea. You know about the Dead Sea? The name already tells you there is no life there. The Dead Sea is the lowest point in the world and also one of the warmest, not maybe the warmest itself, but at the top of being warm, and it's in Israel. And although it's called Dead Sea, there are 2 types of bacteria there, and I knew already a little bit about the life of one of them, because there were 2 professors that were working on them, and we collaborated with them. So I thought, this is a system that is always under pressure. Maybe I can look at their ribosomes. And it worked. And this is the way I started. Now, the ribosomes in the Dead Sea bacteria are also making proteins. They are also reading the messenger RNA instructions. This is the way I started.
So essentially, the bicycle accident was this serendipitous event.
The bicycle accident is the beginning of this project. Not
Again,
only of mine,
please,
of all those that copied from us, which are the rest of the ribosome structures.
But also, it enabled you to actually sort of diverge from what you were working on and just put your mind to something else, and you realized that some other things had to tell you something about the very problem you're working on.
That's correct.
So that's which is great, and it worked out well.
That's correct, and this is the great thing about science.
Yes.
You focus on something, you find something that looks completely different, you find the connection, and if you want, you can work on this connection, which, in my opinion, is a very, very interesting phenomenon.
Yeah. So if you have one advice for the young people, would it be just don't focus
take a bicycle if Don't drive on roads that have holes in them, like mine.
Wear a helmet. If there are any questions that you might have, please use Slido. We have these, not anymore, but go to Slido, #AdaYonath, and we'll get to the questions later. So then you determine the structure of the ribosome, right? And the other groups as well determine the structure of the ribosome about the same time, at the turn of the century, essentially. And then
there were two reasons why it came more or less at the same time. The first reason was the method that we developed for protecting crystals from being bitten by the X-rays. Yeah. And the second, which was, I'll tell you about it in a minute. And the second, that the beam, the X-ray beam, came from synchrotrons. These are big facilities, very, very big rings, hundreds of meters, to produce the X-rays, which had to be at very high intensity and very focused appearance in order to collect the diffraction data from the ribosome crystals. So about preserving. X-rays are also interacting with the bonds and finishing the crystals, or finishing every living system. When you go to take a picture, X-ray picture in a hospital, it's very quick. And the people that are making the picture are standing behind a wall.
Lead curtain, yes.
And only the little place in the body that has to be seen is being irradiated very, very shortly, because X-rays are interfering with bonds in the body, in the crystal, in everywhere. And the intensity that was needed for the ribosome crystals was so high that the crystals that I had disintegrated within the first shot. It means in order to get the whole sphere of diffraction, I needed thousands of crystals. And from each of them, I got less than 2% of the data. So it was, for the ribosomes, this method was useless. It was hardly used for small proteins, but surely not. So after working for 5 years getting crystals, we lost them in 2 minutes. The crystal did not last more than a minute or two.
Essentially evaporated them with the X-ray beams.
Whatever. We are not going to talk about how they were disintegrated, but they did.
Yes.
So can you imagine this? A whole group in Israel. There was meanwhile also a group in Berlin making these crystals, for hundreds of the crystals, and they all got within a few minutes, one after the other. So this was more or less the end of this project.
Yeah.
But I couldn't afford having an end of a project that I was dreaming, working for 4 or 5 years, and getting finished by just X-rays, by the machinery. So I met 2 scientists. I won't go into the meeting. One of them is Ishi Talmon from the Technion, and one is Wao Chu, now in Stanford. They found a way to freeze their crystals. They worked on small molecules, but also wanted to eliminate disruption, to freeze their crystals, and then there was a very much slower destruction. How to freeze crystals of bacteria, of biological materials? They all need to have water, and water becomes ice, and the ice is cutting the crystals. So we couldn't really just freeze. It took us another year to find a way to what we call collecting data at cryo temperatures, minus 200 degrees. But when we found it, crystals did not disintegrate, at least not quickly, after 50–60% of the data, so we could collect. And at that time, I was called the cryo dreamer. I got elevated from just a dreamer to a cryo dreamer. But after one session, it was in Cornell, I think, the rumor became spread, and all synchrotron radiations, there were at that time 6 or 7 in the world, they all wanted to employ this method. Also some universities like MIT. And within 5–6 months, all the crystallographers moved into this method. Now, students don't even know that there is another method, and there are 60,000 structures from the 6 or 7 that were known in the beginning. So not only the bicycle, not only the bears, also the cryocrystallography.
Yeah. So all these are all the good contributions that actually came together and enabled you to
all this has nothing to do with reading the genetic code.
No, that's determining the structure of a ribosome.
Yeah,
but then, for me, if we can just move on slightly, the amazing thing about ribosome is how well conserved the structure is. In every living thing, all the different, slightly different ribosomes that we have, because the sequence of that determines the ribosome might change a little bit during evolution.
Yeah, and also the ribosomes become larger with evolution because they have to interact with the cell. They have to know, the ribosome has to know that in the liver there is a need now for new protein. They have to interact with cellular components.
Yeah, so for example, our ribosomes are bigger than bacterial ribosomes.
Correct.
Right, they're slightly different. But there are parts of the structure of the ribosome that are very well conserved.
Yeah, so the practical parts of the structure, it means the path of the messenger RNA with the information, and more than it, the pocket where the peptide bond is being made, are highly conserved. Especially the pocket. The pocket is almost 98% conserved, maybe even 100, at least in their structure, in their appearance, like a pocket where inside the bond is being made. This is very, very highly conserved. In bacteria, in cockroaches, in flowers, in bears, in human beings, in fish, in birds, same. Even if the whole ribosome structure size is changed by 20%, this pocket is still the same. And this gave us the feeling that the pocket is not affected by evolution.
Which means that it's probably so important that it actually stays the same.
Stay the same, maybe even was before the ribosome.
And I think it actually is something that provides a window or a lens, you can say, on the whole evolution and the origin of life.
Okay.
Because we can trace this essentially 4 billion years ago.
Correct. I'm not sure about billions, but how many billions?
I mean, I don't remember what happened last week, but yes.
That pockets like this were created accidentally. RNA was available, existed before life. So pieces like this became formed probably accidentally. And amino acids started to show up from wherever, I don't know. Those that found themselves into the pocket stood there and made bonds. So we think that this is the origin of life. That's life, the way life started. And when there is one bond, can be second bond and third bond, like now, and small proteins, small, I won't call them proteins, but small peptides were made.
Yeah.
And those that were needed for life in the RNA world, they survive. So they led into the production of more pockets like this, and more pockets that were stabilized or interacting with the small proteins that were made there, the peptides. So this is why we thought that this is origin of life. More than this, since it is so hardly preserved, this pocket in the whole ribosome, it is the highest reserved pieces. We called it the proto-ribosome. It means the beginning of the ribosome. So we had this idea for years, almost 10 years, maybe even more, 12, until we thought maybe we should produce this piece and see whether it can survive. So this took another 5 years. These projects seem to be very simple, but they are difficult to organize, to develop, to design, all these things. Yeah. And then we could show that we can get dimers of 2 pieces that make the pocket. And another 5 years, we could even get peptide bonds in them if we used amino acids. So we have now the production of what we call proto-ribosome, and we think that we understand the origin of life.
I think this is amazing because there's one thing we sort of glanced over. When you have a ribosome, it's not just RNA, it's also complex made with proteins. So essentially, you need proteins to make proteins in our cell.
In order to stabilize
exactly. So it's sort of like the chicken versus the egg problem, right? How do you make proteins to start with so we can continue making proteins?
That's correct. This is chicken and egg, but we didn't answer what was first, the chicken or the egg.
No.
Sorry.
You focused on the RNA
This is our dream.
Exactly, sorry. This remains one of the most profound scientific questions still. What was first, chicken or the egg?
That's correct. But this gives us, and I hope now also you, the notion of what could have been the beginning of life, the origin of life.
It's also because the RNA is such an essential molecule. Because we always thought, the textbook description was that it's essentially a template that keeps information, right? But it also can have catalytic functions. It can have many other
functions. Can have structural functions.
And structural functions. So we can actually work as a sort of proto-enzymes, right? Ribozymes that do something less effectively, less efficiently than enzymes and proteins, but still does that. And what you showed, essentially, is that you can get away with proteins in the beginning and just use just RNA to start essentially bootstrapping the whole world.
Correct.
Which is...
Do you agree, ladies and gentlemen? Or if you don't and you want to start a discussion and tell me how stupid I am, I'll be happy to hear it. Because critique is building.
So since you started with that, I mean, my usual experience is that life experience and scientific existence is actually pretty abysmal, because every day you find out what you don't know. And you have to live with that there are so many things you don't know. I don't know if that's your experience, but things don't work.
You're telling me?
Yeah, I don't. I'm asking.
After so many attempts to understand the world.
Yes. It's my experience that once you actually understand, or you think you understand everything, right, you're done. Because there's not much more you can actually learn at this point. So Yeah, we have lights moving.
Yeah, well, the ribosome has to work.
Yes, exactly. So it's producing proteins right now, probably. But then also...
Sure, in all of us.
All of us are producing proteins all the time. But then also, since we know essentially how it might have started, with the protoribosome bootstrapping the whole world, starting the production of proteins, for example. But then the question is, how did the components of RNA actually appear? Do we know how that can be synthesized in those proto-conditions?
You see, I'm still young,
Yes.
and I still have to answer these questions. I didn't do this yet.
Okay, so that would be...
But if not me, maybe one of you, or many of you.
And also, you sort of alluded to it, but, well, we might know how the proteins actually are produced if we have the building components of proteins and RNA and everything, right?
And the sequence.
And the sequence. But how did we start coding for amino acids? Because that doesn't answer that question.
No,
right?
it's after.
It's after, yes.
It's much after. It's second, third stage.
Right, so how did it appear that now we have a genetic code, that we have 20 amino acids built into proteins, each one of them is coded by several combinations of 3 letters of the genetic alphabet?
I can't answer this. It means I can tell you what I read, but
Yes, exactly.
but I cannot. We still didn't show this.
Yeah.
I'm not sure that we are close to this. So far, given the sequence is known, we don't know how things work.
Exactly. But I think this is actually I think this is great, I have to say, because for me, excellent research just opens more questions than it tends to answer, right? So if you just keep asking the questions all the time, right, and new questions, I mean, you determine the structure of the ribosome and many ribosomes, but then what's next, right? What does it mean?
Go to the hibernating bears. Maybe they will tell you.
Oh, we have plenty of bears in Slovakia, so we can have, every winter we can just go and just collect.
Yeah, it's closer for you than for me.
And it's, yes, for us, I mean, going to the Dead Sea probably might not be, I don't know how the grant agencies would look at that. Let's not test
maybe
them.
if you say that this was the key for ribosome structure,
Yes. But then, essentially, what you did with your protoribosome, you disappointed our biologists because you just showed them, like, look, all life, all biology is just chemistry, right?
Yeah, but chemistry. This is clear.
Thank you. So now you're not making it better.
The biologists don't like it. Yeah. They don't need the genetic code for that. They just don't like it.
Yeah.
I don't want to say all the biologists, but many of them.
Many of them. But then if we move to the RNA world, to the hypothetical world, billions of years ago, when the RNA was the ruling molecule of the whole world, and then there was probably something before that, right? The pre-RNA world, even simpler molecules that led to the evolution of RNA, and then we can say that just all chemistry is just physics?
Well, the physicists would like to
Physicists
say
will like that, yes.
But actually, all physics is chemistry.
Physics is chemistry, yes.
It is connected, but the other way, because physics has nothing to do if there are no atoms.
But they will tell you that if you know all the positions of atoms, you can describe them and write the equations.
And now the physicists were able, last week I think, to show that they can hit bodies in the atmosphere. They can directly hit them and even change their ellipse of motion. So maybe there is some hope to understand other worlds. But I'm not sure there was an RNA world. There was an RNA controlled world or RNA prominent world, but there were other things too.
Yes. And how do you think the basic, we sort of talked about it very briefly, the basic components came into being, essentially? Did they originate on Earth, or did they come from somewhere else, or did they just stop by on a meteorite?
Maybe I need another bicycle accident.
And yet another animal. Maybe lions would be something that might... Oh, they sleep too much.
Sorry, I can't. I can't answer this.
Sorry. It's all right.
But maybe there are other questions that you want me to answer.
Yes, I can take a quick look at Slido. Some of these questions we already started and answered. And I think we will answer some of these questions, but then there's one interesting question that it's very relevant right now. You mentioned that there are thousands or tens of thousands of structures that are solved, right? And then there's this DeepMind project of protein folding. And then, what do you think about that?
So I expected it, therefore, I expected it as a question, therefore I didn't relate to it. But what was shown is that the structure of proteins is determined by the sequence, and the sequence is capable of making interactions within the protein. This is the way that the structure is there. And these interactions have some rules, that they are not everything. It means if I decide to interact with you, it either works or not, depending on several physical, biological conditions. Now, one of the first people that thought about it, his name is John Moult. He came from Oxford, but he was postdocing in Israel. And he is actually the person that helped me in starting crystallography in Israel. Means a lot is his contribution. He is now in Maryland, and he started in '94, I think, like more than 20 years ago, a competition, a world competition, once in 2 years, that he divided the people that wanted to compete to two groups. One group that determined the structure experimentally of a protein that the structure is not known, determined it experimentally from crystals, and one that predicted it from the sequence based on known structures. So the beginning of what is called successful hits was not very high, but I think we're thinking about over 35%, 35, 36%. And this went on to almost 50% after 14 years. It means 7 or even the 8th competition. And more groups were involved in it. Like, I think at the end they had 91 groups and so on. Mm-hmm. And then Google came in.
And they have a lot of data.
And they had lots of data, all the data that was already known, structures known, and the hits of this competition, the competition called CASP. So, and they collaborated, and they wrote a program that takes into account all the data from structures, known structures. Now they have 95, 96% agreement between the structure, the actual structure, and I think it's called the
AlphaFold.
Alpha-1.
Or something like that.
Alpha-1, the structure, the program is called. 95, 96, and they even talk about 98, which I'm not sure they are really right. 98% agreement in predicting structures. But my last interaction with them, my last nailing is the ribosome is too complicated for us. So we still have
some edge.
Something to look for.
Exactly. So, but even their algorithms can work only given the existing huge database of already solved structures.
Correct.
And then
and energetical considerations.
Yes. So it also thanks to you that they can actually succeed.
And they are very, very friendly.
Yes, I know. Of course they are. Why wouldn't that be?
They have a little clip, little video that shows the first time that they put in. I can send you the address, and you can give it to all, which is really very nice, and you can see even how John, that I just mentioned, is talking to them, and they sit near the terminals, near the screens, and show.
Yeah. So these structures that we talk about a lot, they don't only tell us about the origin of life, how well they're conserved, tells us how long they've been developing and evolving, but they also have very practical implications, right? Because these ribosomes are also targets of antibiotics.
Yeah,
and we'll just continue with slowly moving towards the different, more practical things.
Yeah, so there are two paths of practical. One is antibiotics, and second is genetic diseases.
Yes.
So if I just tell about genetic diseases in 2 sentences,
okay.
There are many diseases that are connected to mutations in ribosomal components. Like 25% of the cancers are directly related to mutations in ribosome components. An anemia, which is very known, it's called DBA, has 96 different, has connection to 96 different mutations in ribosomal components. So we are also looking into it now, how these things happen, but much more
immediate impact.
Functional and immediate impact is the antibiotics. So you can imagine that if a bacteria want to be pathogenic, it will develop something specific in one of its important functional sites, and one of them is ribosomes. So actually, 50% of the pathogenic fighting against pathogens, it means antibiotics are targeting the PTC, this pocket, our pocket. So there are several very small differences between bacteria and resistant bacteria at this pocket, which is a target of a lot of antibiotics. So if antibiotic can target only the bacteria and not the patient, it is useful antibiotic.
That might be actually welcome.
Like all the macrolides are targeting the PTC, the pocket, and the difference between them and the resistant bacteria is one nucleotide. One nucleotide gives resistance, full resistance to all macrolides. And understanding this, first it's understanding, but second is maybe could be antibiotics that overcome this resistance. Between bacteria, the bacteria want to live. The pathogens, they want to live. So they make resistance, they produce resistance almost immediately.
Yeah.
And I guess that they produce changes, hundreds or thousands of changes, and those that are resistant, those exist. And those are
that's essentially evolution in real time, right? When you put the antibiotics to bacteria.
Correct. Yeah. And there are resistance to antibiotics in human, in animals, in fish, and so on. So this is one way, and we are also working on it, trying to suggest macrolides or antibiotics that will go into ribosomes of the pathogens, either before or if they cannot make resistance to them. And we are doing it together with several universities like Michigan, but companies don't like it because they know that bacteria want to live and they know that they will have resistance. So there is some economic problems about fighting against resistance for antibiotics. And there is also a new idea, at least our new idea. I think there are some more, but I don't know all about them. And then looking at sites on the pathogens on the periphery, not at the active sites, but at the envelope, that are busy in interacting with cell components and targeting them.
So those are the ones that are less likely to change, because they're also essential for survival.
I don't know, but the bacteria did not understand how important they are, not yet.
They don't want to change it because they don't know about
They still don't know, yeah.
Exactly.
So I do think that 10 years is enough for bacteria to learn.
Yes.
And you know how many bacteria will die until then. I don't even want to think about the number. But in my opinion, 10 years is more or less the time for existing antibiotic resistance. And we found 16 sites like this, so 160 years if we go one by one.
That will be fine.
I think for me it's enough.
Yes. But then...
But it's not, the way I speak is like everything is there, is only to look at it and go on. No, this is not the situation. Yeah. This is still in very initial conditions. We don't know everything about it. And it doesn't say that it will really work. It works in the test tube. But in the patient, I'm not sure. We have to check that.
I would just like to point out that this is yet another example of how understanding of the ribosome, the structure of the ribosome, actually helped us understanding the mechanism of action of antibiotics. Because many of them were developed before we knew the structure of ribosomes.
Sure.
And they worked.
The bacteria did not care what we know.
Exactly. Yeah. Nature doesn't care about what we want. Yeah.
So if I can show, can you see my fingers? If this is the pocket, it's enough to have this as raising antibiotic resistance. Yeah. Just one little change, and thousands, maybe even millions of people suffering either from the disease or really dying because of it, or animals. Resistance to antibiotics is a real big problem in medicine.
Yes, it is, and we see that in hospitals.
It's really real.
All the time. But then, again, so the success of antibiotics is essentially built on because half of antibiotics we know target the ribosome, right? Approximately, and then the success is built on the very fact that the bacterial ribosomes are slightly different than our ribosomes.
Yeah.
So it doesn't, as you said, it doesn't kill us, but it kills.
Correct, but the success is to avoid the little change that I talked about. This example is changing one component that is different between bacteria and human, from bacteria to human, so then it is resistant.
But we also have bacterial chromosomes in us, in mitochondria, right? Essentially, right? So how come the antibiotics actually don't kill those ribosomes and don't kill our mitochondria?
I don't know if they don't kill or do kill.
I hope not.
The mitochondria ribosomes are still not a target. So there is some information.
Maybe because they're well hidden behind the mitochondrial membranes, so the antibiotics cannot get to them.
Yeah, but they have also to go through the cell membrane.
Yeah.
So the antibiotics are going, the antibiotics themselves can penetrate envelopes, cellular envelopes, mitochondrial envelopes. And afterwards, they know their way to their target. They don't sit there in the coffee house and discuss. They just directly go to where they think they want or they are needed. There are hundreds of problems.
Yes, which is great for scientists.
And there are antibiotics that are slightly damaging the carrier. There are antibiotics against pathogens for human and pathogens for animals. It's still open field, let's call it, an open field for many scientists. I'm not even talking about companies.
But you might be, I think you're optimistic that we can actually be able to design, that we can design new antibiotics that will be specifically targeting those sequences or those structures on the bacterial ribosomes.
Semi-optimistic.
Semi-optimistic. Okay, still better than semi-pessimistic. So the glass is half full, right? In your case, literally half full.
Now yours is half empty.
No. Mine is actually almost full, so that's it's better. So once we have these ribosomes, we talked about them, that they're essentially molecular machines, right? I know this is not your area, but what do you think about, let's say, nanoscale machines that people are producing now? Because based on what we know about ribosomes, we can create these really small machines, I mean, essentially nanorobots, that you might imagine might be able...
It's wonderful. It's the future. But how will you tell these nanorobots that a protein is being needed?
So that's another thing that we do not understand. How to start talking to them.
Both you and me and many others don't really understand.
Oh, no. Oh, yes, I agree. 100%.
But once this will be understood, it will be wonderful.
Yeah, because you can imagine if we can actually target them where needed and initiate their action when needed, that would be wonderful.
And
if we can stop them at some point.
The cell that didn't go to university, didn't take classes, will find a way to find them
That's
too.
Trouble after
but
bicycle
maybe
accident.
It will be longer than just biological materials.
That might be one of the approaches that we might actually...
One of the benefits.
Yes.
But although we are not doing it, I'm not doing it, I highly appreciate it.
I know. To me, it still sounds more like a science fiction, although I know there are groups working on it. And when you started, right, you said that the people were sort of making fun of you when you were trying to make crystals, right? And then you had some trouble continuing in your career. No,
the trouble was only
in the beginning.
In production. It means
yes, in production.
If I can create a group, if I can get granted, the laughing of people, I can't care less.
Excellent.
They could laugh. I didn't always understand why they laugh, and I didn't have time to think about it. I had so many problems.
Too much work.
So to think that Mr. X and Mr. Y are making jokes of me.
So what do you think is the main personal trait that you would like to see, not in a scientist, but in any human being?
I don't know about human being in general. It's much too complicated for me. But in science, go after your curiosity. Don't care for anything else. Ask the questions that really, really are interesting for you, and that you know that they have value, not
yes.
Why a flower is red. This is a very good question, but maybe the value is not so high. Maybe it is, I don't know. But find what you're curious about and go after it. And try to, at least to convince yourself.
So, but then, I think, to me, most children are natural scientists because they're curious all the time. They ask questions, they just want to know how things work. Why? And then something happens, right? And in most adults, I'm not looking at you in particular, and anyone, but in most adults, the curiosity sort of dies out sometimes. How do we keep
because
the children...
of other adults.
because of other adults, maybe. So, but as long as we need to live in an environment where there's more people, how do we actually? What would you suggest that we do to keep the curiosity going? Because
I wish I knew.
Yeah.
I wish I knew, but I know that kids are curious. Some are more and some are less. And if curiosity is not killed, is not turned down by adults that's supposed to know everything,
Yeah.
then there is a chance that this curiosity will lead to new knowledge or new interactions or even of new art.
And so I think your curiosity is still going strong. And so what are you working on these days in your lab, in your group?
The proto-ribosome.
The proto-ribosome.
To show how it became ribosome from proto. New antibiotics and the reasons and the appearance of genetic diseases, ribosomal genetic diseases.
So,
I want to tell you that I asked for 3 grants on it and I was kicked out. It's too far.
yes, and if you are declined a grant, then where's the hope for us? Right?
Oh, you're still young.
That is true.
And you did not realize a lot of jokes about you, so you're still, maybe you can convince. But anyway, I think that young people should be encouraged much more than older ones.
Yes.
Because maybe they really have very, very unbelievable fresh ideas. And that's what drives science and life.
Exactly, and I think those ideas that are really out there, they're really high-risk, high-gain ideas, should be encouraged.
Correct.
Especially in the young
This is my
people.
opinion.
I mean, I think, in my opinion, most of them will fail, but those few that will not, that will just make up for that.
Yeah, well, I think that I would be a little bit less critical than you. I won't say fail, but I will say not showing.
Not just exactly.
No, not reaching.
Okay, so you're more optimistic than I am. So it's a better description. Thank you.
Well, I'm just more. It's not very nice, but I'm older.
No, I'm not going to say anything right now. But so let's. I want to just look at some questions. I think we answered most of them. We focused on many of them, and then there are questions about serendipity, how ribosomes came into existence, and sort of. But there's one interesting question that is sort of relevant to the last points in our discussion. Is what do you see about the role of women and the female scientists? How do you? I mean, I'm the wrong person to ask that question, obviously, but that comes from Slido because. So, do you have any opinions on that? I mean, do you think that the situation is changing? Was there any difference in the situation? What are your experiences?
I can answer you what do I think about it.
Yes.
I think that science is gender independent.
Yes.
It's dependent much more of curiosity, and to divide the world to more curious or less according to gender, I'm not sure it will work.
Yes.
The fact that there are less female in science, at least not in science maybe there are more female, but in high levels or in prizes, it's just because female in the past were turned to think that it's not good to be a big boss because one has to be also, a woman has to be more at home or with children and so on. This changes a bit now. Also, there are still places that, I don't want to say pay the men better, but men can ask for better salaries or conditions, and sometimes it works. So this, if it happens, should be changed.
Yes.
But otherwise, because female by the new generations think less that they worth less, they're making it. I'm not sure that there is a gender point.
Thank you. Thank you for that opinion. I do believe that science, as you said, is gender neutral, because nature doesn't care about us. What we think.
In my case, I didn't think about prices or development. I only wanted to work, and they gave me this position that cannot be promoted. This was my best time. And men are not educated this way, so they think about competition, and then science that is really not done only for competition, but for understanding and for shedding light and so on, becomes competition, then it also may fail.
Yeah, because then the goal becomes different goal.
Correct.
Instead of understanding, it's competing.
Competing, promoting, being more recognized, giving prizes like asset.
Yes, and the Asset Foundation, exactly, yes. Which is great, and we're thankful and grateful.
Yes, correct. But if it brings unwanted competition, it is destructive.
Yes.
So the balance has to be kept very carefully.
It cannot be the goal.
It cannot be the goal.
It's one of the...
So all these guys here that didn't get the asset prize, it's still okay.
Yes. And most people will not get it anyway, right? The same applies to Nobel Prizes, by the way, but still.
Yeah, the Nobel Prizes everybody gets.
Thank you. So there's hope.
There is a hope.
But I also I have another question. Why is all of this actually happening? Meaning that we talked about the ribosomes, right? And the ribosomes, we need to make them. We have many of them in our cells. They need energy, right? And I think most of the energy that a cell requires actually goes to producing ribosomes and making proteins.
You know this?
60%, I don't know, 60%, the estimates, right?
Okay.
But essentially,
I didn't know that, but it's good, thank you.
But essentially, then that would mean that we, as every live being, be it a bacteria, be it a plant or a mammal, fish, bear, for example, our existence is just building our ribosomes, essentially. Everything else is just on top of that. So are we here to keep the ribosomes happy? Or are we here just to propagate our genes, the DNA? We didn't even talk about DNA most of the time, because that's just an afterthought, I guess. That's RNA is the most important.
You're looking for the reason that we exist?
I don't know. Well,
I
obviously
think that there is a spiritual reason.
And I thought we exist to propagate our genes and to feed our ribosomes. And actually to keep the membrane potential going in our neurons as well. That's also.
Yeah, we didn't talk about membranes at all.
Yeah, we did not. That's true.
Okay, next year.
So, thank you. Thank you. We are all very grateful that you could have been here with us. Thank you for sharing your thoughts, and thank you for being here. I do hope this was as exciting for our audience as it was for me. Thank you again, and hopefully we'll see you again in Bratislava at some point, perhaps in not too distant future. If not, we will come to the Dead Sea and collect some of the last remaining bacteria.
You should go to the Dead Sea because it's really interesting there. It's really something else, and it's going to die.
And it also gave us big clues about the origin of life, despite its name. So I think that was amazing. Also, I would like to thank all of you for coming, all of those who were watching and listening, those who were watching and listening with understanding, that's even better. So thank you for being here. We thank Asset Science Awards. We thank the Asset Foundation for support. And thank you again. Please come back for the next asset prizes and for the next discussions. And please, everyone, do stay curious. Thank you. This is for you.
It's very important because I'm going to fly.
Going to fly. We just keep it here for you. Thank you. And just we'll keep it away.
But, take it home.
Okay, I will. Thank you. I'll just keep it here.
Thank you.
I will try to get up. Nakatulog. Nakatulog.